Well, I'm glad you joined me for a discussion of Chapter 4 in our microbiology text. We've already had an introduction to microbiology, and now we're going to look at the structure and function of prokaryotic and eukaryotic microbes. Just a micrograph showing some bacteria. Serratia marcescens is an interesting bacteria you'll encounter in lab. It grows differently at different temperatures. It has a bright red color in one temperature and colorless at another temperature. So I think it's bright red at body temperature 37 degrees Celsius, but you'll find out about that in the lab. So prokaryotes are microorganisms that have no nuclei, whereas eukaryotes have nuclei. In a prokaryote, there's no nucleus, but there is a genome. It has to have genes to be able to synthesize proteins that make up the structure of the bacteria. For example, there's a circular chromosome, long raveled up circle of DNA that's all packaged up inside the cell, but not in a nucleus. You may remember also maybe from general biology that in eukaryotes, the DNA is wrapped up around little discs of protein forming like beads on a string kind of arrangement. It's coiled up around histones, clusters of histones. There's no histones in the bacterial genome. There are no organelles, no membrane-bound organelles in a prokaryote. Everything's just kind of floating around in the cytoplasm of the cell. Bacteria have cell walls made up of peptidoglycan. We'll take a closer look at what in the world is that. And it's unique to bacteria. Archaea, which we really won't treat very much, have a different kind of cell wall. They do not have peptidoglycan cell walls. When these things divide, when prokaryotes divide, they tend to enlarge and then break into two symmetrical pieces, binary fission, pop into two daughter cells that look much like the parent cell. Eukaryotes have paired chromosomes within a membrane-enclosed nucleus. There are histones, again, little discs of proteins around which the sections of the DNA are coiled like beads on a string. There's numerous organelles wrapped up in lipid bilayer, the same kind of arrangement of phospholipids as in the cell membrane. And there may be a polysaccharide cell wall, like in plant cells, or no cell wall, like in animal cells. During mitosis, there's actually a mitotic spindle to separate homologous or identical chromosomes that have been produced by replication right before the division of the cells. Here is just a for fun micrograph showing a particular type of bacteria and its flagella. It's an organelle that it has to help it. I guess it's called an organelle. It's a bunch of protein stalks sticking out of the cell that allow it to have motility and drive like a little motorboat through the water. So let's look at some shapes. A bacillus, we're talking now about prokaryotes. Bacteria can be rod-shaped or spherical. Or they can have a curved arrangement. Instead of being a straight rod, they can have curves. So there's three different types of curved bacteria shown here that we'll look at more closely later on. Arrangements of cocci. So if a coccus is a single bacterium, multiple cocci, and the cocci can be found in pairs, in which case the prefix diplo is used, diplococci. And streptococci, this is a long chain of cocci. And these are important for us because often clinically, diagnosis of disease, we may know something about the disease we can observe in the patient, and then looking at the arrangement of bacteria in a stain may quickly allow us to differentiate between a couple of possibilities for which bacteria is causing the disease. So we can have single bacilli. Coccobacilli are like an intermediate between a coccus and a bacillus, kind of a stubby little short bacillus. Vibrio refers to curved cells that they otherwise would be a bacillus, but they have this curve to them, so they have this nickname Vibrio. Sporillum are kind of coil-shaped or spring-shaped bacteria, and they're rigid. They're called sporillum. You have to be very careful to differentiate those from spirochetes. Spirochetes are similarly looking corkscrew-shaped bacteria, but they're flexible, and they can drive through the water with a specialized kind of a flagellum, as we'll see. Kind of cool. So we can have diplococci or diplobacilli, staphylococci, which we'll see in a second. Baby, no. Discuss staphylococci. So sometimes when staph bacteria divide, they remain together each time and form large clusters. And I'm sure you've heard of staph bacteria, Staphylococcus bacteria. That's kind of how they look in clusters. Diplobacilli, streptobacilli, just like streptococcus, you can have streptobacillus. Here's a little schematic diagram of a prokaryote. You can see that it has its chromosome. It's actually a circular arrangement of DNA, but it's very long and it's all raveled up into a blob. And just because there's no membrane-bound organelles, it doesn't mean that the cytoplasm is homogeneous. The region around the DNA is unique and has different proteins than found elsewhere in the cytoplasm. It's called the nucleoid. So there's also lots of ribosomes you see floating around in there. They're called 70S ribosomes for their sort of density. They're a certain type and shape of ribosome that's different from the ribosome in eukaryotic cells, as we'll see later on. There's a cell wall around this bacterium, and then that's the brown. We'll take a closer look at that. In this case, not always, but in this case, there's a capsule, a polysaccharide shell around the outside of that. Optionally, some bacteria have flagella sticking out. These are for they can actually rotate and move the bacteria, allow them to have motility. There are pili or pili and fimbria. That's these hair-like structures sticking out from the surface. Here's a pilus. Here's a bunch of fimbria. As shown in this little paragraph, not all bacteria have all of these structures, and so the structures in red are common to all bacteria, but some have inclusions, specialized containers, essentially, or dense packaging of certain materials. A plasmid is like a little parasitic piece of DNA, a little circular piece of DNA much smaller than the chromosome, which will divide along with the chromosome within the cytoplasm of the bacteria. And actually, they can divide many times. There can be quite a few plasmids inside of a bacterium. But interestingly, plasmids can, number one, be shared between bacteria, which is a kind of a big problem. And number two, because of the problem comes up when the plasmids carry genes that allow the bacteria to cause disease. So this is like a little rider inside the bacteria, but it may contribute very much to the properties of the bacteria. The glycocalyx, this capsule on the outside, can be either sticky or it can be neatly organized, so-called capsules, just polysaccharides, chains of sugars attached together. And sometimes it allows the cells to attach onto something, but also capsules around bacteria sometimes allow them to circumvent phagocytosis by macrophages and so forth, so they can't be eaten up and killed. They slip right off. The macrophage can't get a hold of the bacterium, and so some bacteria cause disease specifically because they have this capsule on the surface. And here's a little micrograph of a bacterium, actually a pair of bacteria, Diplococcus, with a large, thick capsule on the outside, which again would make this Streptococcus pneumoniae is a disease-causing bacteria that causes pneumonia. We have a vaccine for that. It's a very bad actor, and it has a capsule that gives it its disease-causing capabilities. Famously, experiments were done showing that this exact strain of bacteria, without the gene that allows it to make a capsule, is harmless and not disease-causing. As soon as it has that capsule gene, boom, causes disease. Flagella, again, are strands of proteins, a stalk of protein attached to the bacterium It allows it to have motility. Let's take a look at a and so we can see here this stalk protein filamentous stalk of the actual body of the flagellum. There's a so-called basal body that anchors the thing with inside of the cell wall and in the cell membrane, and then there's this so-called hook, which is what generates the turning, the spinning of the flagellum that allows the bacteria to actually move. This is a gram-positive bacteria, and we haven't really talked about gram-positive and gram-negative. Here's a gram-negative cell. It has an extra membrane, has two lipid bilayers. This outer membrane is just shown in this picture, but it's a very similar thing that's happening here. We have this flagellum anchored into the cell wall and the cell membrane, and it sticks out and allows the cell to move as it spins like a little propeller. Arrangements of flagella have special names. Paratrichous arrangement means that the flagella are all over the surface of the bacterium. Monotrichous means a single flagellum. Lophotrichous means a tuft or a clump of flagella. It can be either polar on the ends or not. There could be one on each end or one on just on tuft on one end. Amphitrichous means on opposite ends. So in this case, there would be a flagellum on each end of this thing. Ampha meaning both, or I have a visitor. Let's see if you can see this visitor down here. There we go. Yeah, got a little visitor coming in. That's Whiskey. Stands for whiskers. He's got white whiskers. All right, motile cells. So what do cells do with their flagella? The movement is not random entirely. When the flagella rotate clockwise, they cause the cell to drive along in a straight line, but then they can switch direction of rotation, and then they expand out from the surface of the bacterium and cause it to move in a random tumbling fashion, and then they switch back to clockwise motion and allow the bacterium to travel in a straight line. And by switching between running and tumbling, the bacteria can move all around in its environment until eventually it finds something tasty that it's interested in having, some nutrient or something like that, and then it will start moving specifically in the direction of the higher concentration of that nutrient. So it's a way it can swim around and find a place where it can have a good habitat. Axial filaments are a special kind of flagella in spirochetes. Remember those corkscrew-looking bacteria? And here we see a spirochete with its axial filament. It's a flagellum that's wrapped around the surface of the cell, and then there's an outer sheath around that. The white lining is just an outer sheath around the flagellum, and the flagellum is anchored at one end, and when it rotates, it causes the bacteria to kind of swim along like a drill in a corkscrew fashion through the water. And here's a little cartoon diagram of this axial filament on a spirochete bacteria. And spirochetes are troublesome bacteria that cause things like syphilis and Lyme disease. Fimbria and pili. Fimbria are protein stalks that allow attachment onto surfaces. Bacteria may be found, for example, attached to an organ in your body, or to a pipe in the water system. So there's some fimbria sticking out of this bacterium and allow them to stick to things. Pili will have multiple possible functions, but one important function that we'll be talking about is the ability of pili is like a little tube, and bacteria can transfer DNA from one cell to another through the pilus. And I was mentioning earlier about sharing DNA from plasmids, genes sometimes that can cause disease. Bacteria can share disease-causing genes, unfortunately, right within the same generation of bacteria by forming a pilus and connecting two bacteria together and then transferring some of that DNA across. The cell wall of bacteria is made up of peptidoglycan. Let's take a look at what that is. So I'm going to skip right to a slide showing peptidoglycan. We can see the cell wall here drawn in this cartoon in yellow, and it looks kind of like this. It's like a mesh or a grid of sugar chains. Polysaccharides are these brown taffy looking things, and they're made up of two alternating units of two different sugars, N-acetylglucosamine and N-acetylmuramic acid. You don't need to memorize those at all. Nag and NAM is not bad to know, but you don't need to worry about the chemical structure. But anyway, alternating presence of those monomers form these chains, and then that's the carbohydrate backbone, and then they're attached together by peptide linkers. And so peptides again are like mini proteins, and so that forms like a screen or a structure of sugar and protein that is the cell wall, peptidoglycan. So in a gram-positive cell, it's very thick. And here's the multi-layered peptidoglycan wall of a gram-positive bacterium. And we can see that there's some proteins attached within the cell wall called teichoic acids. And the reason we bring those up is because teichoic acids are often unique to a particular species or strain of bacteria. And so using antibodies, we can identify this precise strain of bacteria that's causing an infection by the teichoic acids that are present. Lipoteichoic acids are just ones that are anchored right down to the cell membrane of the bacterium. So that's the thick peptidoglycan cell wall of a gram-positive bacterium. Gram-negative bacteria, on the other hand, have a thin peptidoglycan cell wall, and the outer membrane, you'll see in a second, has that outer lipid bilayer, has a bunch of polysaccharides sticking out, which are extremely nasty. It causes massive inflammation in animals. The lipid A specifically causes severe inflammation, and attached to that is an O polysaccharide. So let's take a look at a gram-negative cell. Down here we see the inner membrane, analogous to a gram-positive cell, a thin peptidoglycan cell wall, and then an outer membrane with these lipid A in there. You see the blue things are the lipid A, separate from the typical phospholipids, and attached to that is the O polysaccharide. The O polysaccharide, it turns out, is good for identifying certain strains of gram-negative bacteria. These are nasty disease-causing guys. They're a little bit more resistant to antibiotics and so forth with this outer membrane to protect them. They have pores in the outer membrane to allow substances to diffuse down in. We have a barking problem here. Sorry about that distraction. Great little fellows, but they do get barky at this time of day because they're hoping for some supper. So anyway, pores, there's porins, pore proteins in the outer membrane to allow nutrients to diffuse down to the surface where they can be transported into the cytoplasm of the cell and so forth, and secretions from the cell can escape out into the surrounding water. So gram-negative bacteria have this terrible lipopolysaccharide on the surface, which is very, very harmful and disease-causing and inflammatory to animal cells. Let's talk about how you can tell the difference. One of the first things to do when you're trying to identify a bacterium in the laboratory is gram stain it. Find out if it's gram-positive or gram-negative, and here's what we're going to do. We're going to use a crystal violet stain. It's a basic dye that we learned about before, and we're going to let that penetrate into the cells. All bacteria will become purple colored with this dye. Then we're going to add some iodine. Iodine will combine with the crystal violet inside the cell and clump up and form large particles. Then we're going to use a decolorizing agent. We're going to take some alcohol and immerse the bacteria in that, or splash some on there in our slide where we're staining it. And it's going to, number one, dehydrate the cells. They kind of contract, and it makes the gram-positive cell wall even more densely packed and tight. And it's going to trap those crystal violet iodine particles inside the cells that can't get out. Whereas in gram-negative cells, the alcohol causes the dye to wash right out of the cells and they become colorless. And finally, we will counterstain the gram-negative cells so that we can see them. So gram stain can help us identify gram-positive versus gram-negative bacteria. Here's a slide that maybe kind of got out of order. Gram-negative bacteria are troublesome because they're harder to kill than gram-positive bacteria, depending on the antibiotics that you have. All right. All right, so how do we do a gram stain? Well, first thing we got to do is get something that we can stain. So we form a smear of bacteria, a thin film of bacteria on the surface of a slide, and let it dry. And then once it's completely dry, We pass it through the flame of a Bunsen burner 1 or 2 times, and that evaporates the last bit of water between the smear and the glass, and the smear will then adhere to the glass, stick right on. Then we can do our staining without washing away the bacteria. So first we're going to add the primary stain, it's called. That's that crystal violet that's going to stain all bacterial cells purple. Then we're going to add the mordant. It's going to make the dye particles larger, so we call it a mordant. It enlarges something. So it's going to, all the cells are still purple. All the cells in our color, no matter whether they're gram-positive or negative. But now there's big dye particles inside. We'll use a decolorizing agent, alcohol acetone mixture, and it'll wash the dye out of gram-negative cells. They have that thin peptidoglycan cell wall, and the dye will come right out. And then, but the gram-positive cells will remain purple. The stain will be trapped. And then we'll counter stain so that we can see the gram-negative cells. If we didn't do that, we wouldn't know if they were even there or not. We use saffron, and it's just a traditionally used counter stain. It's a red dye instead of purple. And we wind up with this. We'll wind up with, in the end, pink gram-negative cells and purple gram-positive cells. This is just a little cartoon flow chart of what's happening in each step. And you should be able to, in your mind, recapitulate each of these steps. You should be able to write down a list, right from scratch on a piece of paper, what's happening to both gram-negative and gram-positive bacteria in each step of the way. And I'll ask you questions like, what would happen if someone stopped the treatment point, this gram staining process at the iodine step? What would the gram-negative and gram-positive cells each look like? Or what if we stopped this process right at the decolorization step? Right after decolorizing, what would the gram-negative cells look like? What would the gram-positive cells look like? And so forth. I want you to thoroughly understand this method. It's super important for how microbiologists proceed in breaking down, classifying microorganisms that are of interest as a first step in identifying them. And here's a nice gram stain showing some, looks like diplobacillus, or maybe it was streptobacillus, and some other gram-negative bacilli in this picture. There's actually some gram-positive cocci. Looks like streptococci right there, little clusters of cocci. There's another important stain that differentiates specific types of bacteria. 2 genera of bacteria, but we're going to stick with Mycobacterium as a genus of interest, have a waxy lipid in the cell wall called mycolic acid, and they won't stain by the typical gram staining method. We need to use a more violent step for staining and a different way of destaining in order to identify them. So what we're going to do is use a primary stain of carbolfuchsin. We're going to heat the bacteria in the presence of the stain very, very hot. And that's going to drive the stain into this waxy lipid cell wall. And then we're going to use, again, a very aggressive decolorizing step, an acid alcohol wash, to get the dye out of all other types of cells in the genus Mycobacterium. Bacteria will remain colored. Let me show you what I mean. So first we do the primary stain. All cells are stained red. We heat with this carbolfuchsin. Then we use the decolorizing agent, and Mycobacterium bacteria remain red, and all other species or genre of bacteria are now colorless. And then we'll counter stain again so we can see the other bacteria with methylene blue. This is traditionally used. You could use any counter stain. It's not red, but we use methylene blue typically. So here we see some Mycobacterium tuberculosis, possibly, for example. In this case, it's Mycobacterium bovis. I see the label on there now, but it's another species. That's not a disease-causing species in humans, but Mycobacterium tuberculosis and leprae are the 2 important species for our purposes that cause disease in humans. And you can see some streptococcus here that are gram-positive, but in this case it's not. They're purple because they were stained with methylene blue, counter-stained with methylene blue. And here again you can see some Mycobacterium tuberculosis within a sample of probably some sputum from a patient's respiratory tract who has TB, tuberculosis. Mycoplasmas are bacteria that don't have a cell wall. They're unique in that way. They're tiny bacteria that don't have a cell wall. Most bacteria have a cell wall, and if they didn't have the cell wall, they would burst. Bacteria don't usually have any way of regulating their cell volume. They just have a cell wall and they fill up with water like a little bullet. There's a great deal of pressure inside there. The cell wall keeps them from bursting, so they don't need to worry about water volume or anything like that. But these cells don't have a cell wall. They can cause a type of pneumonia. Archaea, again, we're not really going to really interest ourselves in archaea very much, but they have a different kind of cell wall altogether from peptidoglycan. One way to tackle a disease-causing microorganism, or bacterium, I should say, is to destroy a cell wall. And lysozyme is an enzyme that's found in body fluids such as tear fluid and mucus. Lysozyme starts to break down bacteria and it helps eliminate the possibility, reduce the likelihood of infections by exposure to bacteria in our eyes and in our mucous membranes, for example. Penicillin is a very, very familiar antibiotic, and the way it works is as bacteria are dividing, they can't produce their cell wall. Their peptidoglycan cell wall production is inhibited in part, and so their cell walls aren't properly constructed, and they begin to swell, and eventually they'll burst, and the cells won't be able to multiply. So that's how penicillin stops infections with bacteria. If a cell wall is destroyed, the remaining bacterial cell with just a cytoplasmic membrane around it is called a protoplast, and it may eventually again swell up and burst. All right, let's stop there, and we'll take up and form a part 2 of this chapter so we don't make too long-winded of a recording. So I'm going to stop right here, and then we'll pick up again and do part 2 of chapter 4 and learn a little bit more about different types of microorganisms, eukaryotes and prokaryotes, particularly eukaryotes next.